Hello, everyone. My name is Sashank Reddy, and I am one of the founders of LifeSprout. I'm joined here today by my friend and colleague and our executive chairman, Adam Gridley. And together, we're delighted to tell you about our company at the Alliance for Regenerative Medicine. As outlined on this slide, LifeSprout sets out to develop revolutionary products for tissue replacement and regeneration. We're excited to unpack that story for you today. To summarize, LifeSprout is developing biomimetic and biostimulatory materials that were originally developed in the laboratory of Dr. Haichuan Mao, an internationally regarded thought leader in nanomaterials at Johns Hopkins. Those technologies were exclusively licensed into our company, and over the last few years, we have turned them into a compelling suite of products for patients with aesthetic and reconstructive needs. We've recently closed a significant round of venture financing that will enable those products to enter clinical trials and will enable us to build a pipeline rather, of technologies behind them and products behind them to enable regenerative cell therapy applications. And that's what we wish to tell you about in the next few minutes. Now, the story begins with us, as it always does, with patients. When Haichuan and I first thought about the state of affairs in the reconstruction of soft tissue defects, and soft tissues are things like skin, fat, muscle, tissues that define the body's form and in many cases contribute critically to function, we felt that the state of affairs was really wanting in our ability to naturally restore these soft tissues without imposing much of a burden on patients. Now, soft tissue defects are very common. They can result from congenital malformation or inflammatory diseases, from serious medical conditions, as you see on this young girl on the left. And they can also result from just the aging of the body itself or the face, where the deficiency and atrophy of soft tissues leads to a hollowed out and more skeletonized appearance. Whatever the cause of these soft tissue defects, we felt no good products that enabled immediate restoration of that volume while encouraging regeneration over time. So to look at the state of affairs when we set out to tackle this problem, there were things like medical devices such as dermal fillers that are illustrated on the left here. These devices were suitable for filling and are suitable for filling relatively small soft tissue defects, yet they don't last, leaving nothing much in their wake and requiring repeat procedures every year or more. And on the other extreme, we have surgical procedures like lipotransfer or free tissue transfer that are illustrated on the right. Now, while these are suitable for larger defects, and while they can in some cases provide more natural restoration and lasting restoration, they have the significant drawback of requiring surgery with all its attendant inconveniences and potential complications. What we wanted to do in the inception of LifeSprout was build a device and a set of technologies, a set of products that really spanned these two things. An off-the-shelf product that nonetheless behaved and performed very much like the body's own soft tissues. And this is indeed what we've come up with, building on the core inventions from Dr. Mao. We've made a biomimetic, biodegradable material that is easily injectable in the office through clinical grade syringes without the need for anesthesia. It immediately fills missing soft tissue volume, and as you'll see, it encourages regeneration over time. Now, when I say it looks and performs like living tissue, some of that is illustrated on this slide. What we're showing you in the upper panels are scanning electron micrographs of our regenerative matrix. That's the term we've given to our platform technology, compared to an electron micrograph of native fat tissue. And I think the architectural similarities between these two are striking and readily apparent. And this is despite the fact that the regenerative matrix is completely synthetic and chemically defined. We know exactly what's going in there. It's the product of engineered synthetic chemistry. There are no cell or animal-derived materials as part of the regenerative matrix. And not only does it look like native tissue with that open porous structure that you see here, it also feels like native tissue. When you bounce your finger on the regenerative matrix on the left here, it feels very similar to the native fat, freshly prepared fat on the right. And not only does it look and feel like living tissue, when you implant the regenerative matrix, we also see a much more natural tissue integration and encouragement of regeneration than we see with existing biomaterials. So here we're showing you the results of a subcutaneous implantation in rodents, though we've carried this on to other animal models as well. And you can see a robust vascular and cellular ingrowth into the lumina product. That's the term we have. That's the name we've given for the first aesthetic filler to emerge from this platform technology. So many more blood vessels and cells emerging throughout the material compared to standard hyaluronic acid biomaterials on the right. And again, that is enabled by the open porous structure of the Lumina product and of the regenerative matrix platform. As mentioned, the first product from that platform is this revolutionary regenerative dermal filler, which we intend to take into the clinic in the nasolabial fold and mid-face applications starting just this fall. It's an in-office injectable procedure, just like other dermal fillers, and it is reversible due to its principal component of hyaluronic acid. Nonetheless, because of this unique structure that Hai Chuan has been able to engineer into it and the team behind him, this hyaluronic acid-based filler is able to do things that no other hyaluronic acid filler can do, including the regeneration that you saw on the previous slides. It's also illustrated here. When we've put this material into animal models, we see a nice integration, as is illustrated both by the MRI imaging here, the non-invasive imaging, as well as by histology. What you see with our material is an outside-in remodeling of the material and natural tissue ingrowth confined to the space where the filler was injected. And that is in distinction to a market-leading hyaluronic acid-based filler, which is just an inert biomaterial. It fills space, and then as it's resorbed over time, leaves very little tissue or regeneration in its wake. We feel that this core virtue, the ability to immediately fill volume while encouraging regeneration over time, is going to define the next set of aesthetic filler products. And those same virtues, as we'll see in a moment, are important for enabling regenerative cell therapy programs. This is our pipeline. We're, as I mentioned, entering the clinic with the first aesthetic fillers in the coming weeks and months here. And then we wish to build upon that platform with next generation products enabling autologous cell therapies. Now, it's these cell therapy programs that are particularly exciting to me as a scientific founder of the company. The same core virtues, the encouragement of vascular ingrowth, the biomimetic structure of the material, the ability to encourage cell attachment and growth within the material. These same virtues, while they're important in the standalone filler application, they really shine when you think about combining these materials with either autologous or allogeneic cells. The first applications we're contemplating are to use the regenerative matrix in combination with the patient's own adipocytes and adipose-derived stem cells to enable natural, large volume tissue restoration for both facial applications in orphan conditions, as you see in this child above, as well as for aesthetic and reconstructive applications throughout the body. And we aim to take it even beyond those applications, as you'll see in the next slide. This just illustrates the fact that when we embed stem cells within the regenerative matrix, that open porous structure encourages those cells to survive and spread out throughout the material and still confined within the material. Whereas hydrogels, like hyaluronic acid-based hydrogels, of a similar stiffness, because they're so densely cross-linked in order to give them that integrity, these hydrogels do not encourage that natural integration, survival, and spread of the tissues, nor of the cells rather, nor when we implant them into experimental animals, do those materials encourage the vascular ingrowth that is critical for long-term survival and that our material provides. Now, we've published on this. The antecedent nanomaterials were published in Science Translational Medicine just last year, demonstrating their virtues in the reconstruction of small soft tissue defects. And we're building upon that work, as I mentioned, with the incorporation of additional regenerative cell therapies in cardiomyocytes, in neurons for heart and nerve repair, respectively, and in an area I'm particularly excited about, in engineered T-cell therapies for both autoimmune and oncologic conditions, where the regenerative matrix not only promotes survival, but can also be used to confer instruction upon the cells that we are incorporating. And we've manufactured these advanced materials both in our in-house facility in Baltimore, as well as with our excellent CRO, CMO partner Bioserve in California, where the final fill and finish of our materials is currently effected. And these materials are, as I mentioned, going to be entering into clinical trials, these first products in the coming weeks and months here. We've been very fortunate to be supported by a great group of investors and partners, recently concluding a fairly substantial investment round, equity round, that will enable us to take these products into clinical development and to build on that pipeline of regenerative cell therapy programs. Now, of course, none of this work would be possible without an exceptional team. I've already mentioned Dr. Haichuan Mao, who you see in the bottom there, an incredible visionary thought leader who's been a fount of so many ideas for the company, and he is complemented by exceptional folks on the business side and on the regulatory side and on the clinical side as well, many of whom are highlighted on this slide. So with that, I just want to summarize, and again, I'm so grateful for the opportunity to present at the Alliance for Regenerative Medicine today. So we've developed this scalable, versatile solution for tissue restoration built on a powerful biomaterials platform that grew out of Johns Hopkins University and has been built upon by our company. We've used that to develop a suite of products addressing needs in both the self-pay market and in very large reconstructive markets as well. And we aim to bring additional programs in cell therapy to the fore in the coming years, potentially in partnership with some of you. So with that, let me thank you all for your attention. Everybody stay healthy. Have a great day.